Hi, Resia.
Hi, it was so nice speaking to you.
It's great to speak with you. So Resia is in South Africa right now, and she's a researcher whose work I've been interested in for years. She studies bacteria in blood and their activity and what they do in a range of chronic inflammatory conditions. Resia, will you introduce yourself quickly? Just give a little bit of your background.
Yes, I'm from the University of Stellenbosch. I've been here for about 4 years. Previous to that, I was at the University of Pretoria. I'm currently the head of the Department of the Physiological Sciences, and I do research in coagulation and hematology.
Great, very cool. All right, so I'll just hit you up with some questions. Some of these, I've read a lot of your papers, and you obviously, and we've talked before, but some of these, are questions that even I am excited for you to clarify. So, at this point, you've published dozens of papers that show how bacteria are capable of persisting in human tissue and blood, especially the blood, and how they can contribute to inflammatory processes in a range of chronic conditions. And what I'd like to know is what got you interested in studying that topic in the first place?
So over the past years, I've been studying blood coagulation, as well as the structure of red blood cells and platelets in various inflammatory conditions, and particularly looking at the presence of the various dysregulated and circulating inflammatory biomarkers and how they might impact the clotting cascade, as well as cellular function. And in our lab, we use various techniques, including the thromboelastograph, which is another word for it's the TEG or the TEG, which is a point of care device, and it gives clinicians typically a quick idea of blood clotting or blood lysis, clot lysis. I also use the scanning electron microscope and the confocal microscope, as well as ELISAs and flow cytometry. So it all began while we were doing all of these studies during two simultaneous MEC degrees, two students working on the MECs, and they were looking at blood samples from patients with Parkinson's disease as well as Alzheimer's disease, and obviously also comparing it then to healthy age-matched controls. And then we stumbled across the idea that there might be bacteria in blood samples. So how it happened is every morning while we're doing our research, we would typically have some coffee around about 7 o'clock in the tea room, and then everyone will come and sit down and discuss what was happening in their lives. And then inevitably, it will always, almost always just end in in-depth discussion on what's happening in the research and in the lab with every student. And these two students in particular, working on the Parkinson's and the Alzheimer's disease, have been struggling at that stage for a while to get proper, in their mind, proper whole blood samples, which were contamination-free in their disease samples. So when we looked at control samples, or when they looked at their control samples, perfect, nothing wrong with it. And however, they meticulously prepared their samples, aseptically drawing blood, the lab methods, looking at how possibly where the contamination could have been coming from, where they thought it was contamination. We stumbled upon the idea that whatever we did, how aseptic we work, we still found these bacteria in our blood samples from the Parkinson's and Alzheimer's disease samples. So we carrying on with this research, we then discovered that there were bacteria in the samples. And if you look at figure 1, which is a STEM micrograph that we eventually published in FENS in 2015, this discovery followed from there. So as I mentioned, we thought first of all, the first thing is contamination. When we tried to clear that and decided that it wasn't contamination, we started looking a little bit further on where these bacteria could come from. And at that stage, I had a postdoc working in my laboratory and looking at the gut microbiome and how it might influence inflammation. At that stage, I was also already working with Douglas Kell, that you know well, on iron dysregulation, and we were working in systemic inflammation. In that stage, he was still at University of Manchester. And then after discussion on how possibly bacteria could enter into the blood system, we then drew figure 2, which possibly could explain our hypothesis. And it was also during that time when we actually came across your paper, Proal et al. In Discovery Medicine in 2014, when you talked about inflammatory disease and the human microbiome. So this was one of the first papers we looked at when we were trying to figure out what is happening in our own samples. So if we look at figure 2, that figure discusses how we thought at that stage possibly entry of bacteria into the blood system, where possibly there might be gut dysbiosis, and then the bacteria might enter via gut barriers that might be compromised, and then where the systemic immune system barriers might also be compromised. And then Douglas coined the term atopobiosis, dysbiosis being from the gut, and then atopobiosis being bacteria in places where they're not supposed to be. And then the whole idea started developing on how these bacteria and the blood microbiome might in fact be implicated in non-communicable diseases as well as in communicable diseases. So while myself and Doug were further discussing this phenomenon of the microbiome, reading your papers and other papers and many other Skype sessions with people, we came to the conclusion that our discovery definitely was not contamination, because we really tried to work aseptically. So when we thought about the gut dysbiosis process, Doug, with his experience in systems biology and also environmental microbiology, we thought that possibly the same phenomenon could happen in the blood and in the body as what is typically known to happen in the environment, where bacteria may be present but not multiplying. And examples are during drought or when the conditions aren't optimal for them to prosper. And then this could possibly also happen inside the body. And then we formulated our bacterial dormancy hypothesis that is shown in figure 3. So this is just, obviously many of your listeners and researchers will know about the various microbial states, but this is just quick. As we know, there are possibly 2 culturable states in bacteria. There's a viable one where you can culture the bacteria, it's observable, bacteria can replicate. And then you might have a dormant state where it's not obviously viable, but it is also not non-viable, but it's not replicating. And then obviously the 3rd state would be a totally non-viable state. So we thought that bacteria entering the system might be able to go into this dormant state, which is when they can be resuscitated when the conditions inside the body is well. And therefore then we formulated our iron dysregulation and dormant microbes hypothesis when we further studied the interactions between bacteria and probably the inflammagens, which might be the key inflammatory biomarkers in circulation and also in the brain.
Wow, Resia, that is awesome. I'm so glad that our paper played a role in you guys moving forward. That's awesome. I kind of knew that, but it's amazing to hear it. And yeah, that's it's interesting because at the time, I think when I was writing that paper, you know, blood microbiome was considered not to be a thing. So your work helped me better understand the actual way in which bacteria can persist in blood and be able to explain it. So it goes back and forth where your work helped me and vice versa, which is cool. So yeah, I guess when it comes to this, you've done so much work that I wonder if there's a moment or a discovery or a particular person that really stands out to you. Or is that already kind of part of the story with Doug?
I think it was part of the story with Doug, and I think the thing that stood out the most was the fact that we really tried our best not to to find out why we were getting possibly contamination. So I think the thing that came most to mind that we really thought, well, now it must be so, is when we found time and time again, looking at controls, nothing. Looking at you know, the disease, we did find these bacteria. So I think that was the most important thing. And obviously Doug played a crucial role here because of his massive amount of information that he has and his experience and his knowledge of the literature.
Yeah, definitely. It's really interesting. It's almost like you guys kept trying to prove yourself wrong. And by doing that, kept finding interesting things. So that's a pretty cool trend. All right. Well, yeah, let's actually get into then the chronic conditions that you've studied. And you found that bacteria and their inflammagens, that's a word you use in the paper, which are sort of the byproducts or a lot of the proteins and metabolites that these bacteria express when they're in a more active form, that they can contribute to disease processes, especially Alzheimer's and Parkinson's. Can you explain a little bit more about that?
Yes, so we have studied numerous conditions, both systemic and neuroinflammatory conditions, and including obviously Parkinson's and Alzheimer's, where it all started. But we also have looked at rheumatoid arthritis, psoriasis, type 2 diabetes. We have done lots of work on that, as well as hemochromatosis. And then colorectal cancer is also one of the cancers that we've recently been looking at, and then also HIV. So central to all of these inflammatory conditions, we think lies that there's a dysregulation of the inflammatory biomarkers, including iron, and then gut dysbiosis is something that is found in all of these systemic inflammatory conditions, as well as the neuroinflammation. And then, interestingly enough, also of lately, we've been looking closely at periodontitis and gingivitis. And we believe that that plays a crucial role in blood hypercoagulation in all of these conditions. That is one of the hallmarks of these conditions, is a hypercoagulable state. So if you look at figure 4, that is an explanation that we try to show all of the factors involved in, for example, Parkinson's disease. So if you just take that figure as what's happening in general, we know that all of these conditions have a genetic predisposition that might be one of the underlying causative factors in the disease progression. But we also know that environmental factors plays a very big role in the development of these conditions, which include the gut dysbiosis and then periodontitis and gingivitis that I've just mentioned. And both of these processes allow for bacteria to enter, going into dormancy, but then also a fundamental part of this whole process is to shed these inflammagens from the bacterial walls like LPS, and also other products like proteases. So one of the prominent periodontitis bacteria is Porphyromonas gingivalis, and that is well known to have, obviously, its LPSs, but also gingipains, which is a protease, which might also play a very important role in the inflammagens that's in circulation. All of these inflammagens then allow the body to react towards it, and then with an increase circulating inflammatory biomarkers, including the interleukins, as well as, for example, iron. And when these are in circulation, you will find that they can interact directly with platelets and with red blood cells, the membrane receptors. And in that figure, I show just a few pathways, and I will be showing later some more pathways, that platelets can directly be affected by all of these circulating inflammatory biomarkers, the interleukins, but also LPS and gingipains, which can bind to the receptors and activate them. And not only platelets and red blood cells, but also endothelial cells, and also fibrinogen, which is the clotting protein. So all of these processes, together with the genetic predisposition, is factors that might play in to cause and to help in the development of Parkinson's disease, as well as Alzheimer's. So it was during this process in our research that when we looked closer at coagulation, and particularly at fibrinogen, which is the clotting plasma protein, we discovered that during inflammation and in the presence of all of these circulating biomarkers, the interleukins, cytokines, as well as the bacterial wall products and proteases, it can change the biochemistry of the fibrinogen molecule, where the fibrin is changed to form an amyloid structure. And this we have also been studying with various fluorescent amyloid markers using the confocal microscope as well as fluorescent microscopy. An example of that is seen in figure 5. So in figure 5, we show at the top 3 micrographs is typically amyloid found in a healthy aged individual who obviously will have some amyloid misfolding protein in the clots, the fibrin clots. And then the 2 rows, the bottom 2 rows, are from 2 Parkinson's patients, where we studied various, the blue, the red, and the green, of various fluorescent markers for amyloid. Now, the green one is the one that everyone knows, theoflavin T, or THT, that has been known to show amyloid in brains of Alzheimer's patients, so the actual amyloid plaques. So we were then the first to discover that protein in blood, the plasma protein fibrinogen, can actually fold in an amyloid state. Interestingly, when we used our purified fibrinogen model, commercial fibrinogen, or if we used platelet-poor plasma from healthy individuals, as I showed in figure 5, with healthy individuals, very little signal is present. However, if we use these 2 models in the laboratory, we can actually also induce amyloid formation in these 2 models when we add bacterial inflammagens like LPS. And after searching the literature, we actually discovered that there are a vast number of papers that has been discussing over the years the role of bacteria and LPS in the development in Alzheimer's and Parkinson's. And in support of this finding of amyloid and the role of bacteria in development of Alzheimer's and Parkinson's, there was a very nice paper in 2019 by Duminy et al. In Science Advances, where they actually found the bacterial proteases from Porphyromonas gingivalis, the ginger pains that I just previously mentioned, inside the brains of Alzheimer's patients. And so that was just such a nice discovery of theirs that supported what we've been saying all along, is that these inflammagens might enter via dysbiosis or via the mouth, gingivitis and periodontitis. And so in the meantime, just after Duminy published this paper, I had some samples, some Parkinson's blood samples in the laboratory. And then we thought, well, if it's in Alzheimer's brains, it might be present in the blood of the Parkinson's patients on the way to the brain. And so we published a paper in Frontiers to show later in 2019 that the protease gingipains were actually inside the blood samples of Parkinson's individuals.
Yes, I've read that paper. It's so cool. It's so cool to have seen your work come together, because I agree that I've followed your work for a while, and you were the first team to put on my radar that amyloid or alpha-synuclein in Parkinson's could be related to bacterial activity, and also the activity of what bacteria produce in their cell wall components, right? And then Rob Moyer at Harvard, who I also became friends with, he, as you know, put forward his own work showing that in the Alzheimer's brain, amyloid seems to act as an antimicrobial peptide that also does reform in response to bacteria and in his case, viruses like the herpes viruses. And then, like you said, that study that came out, which showed that P. gingivalis might also be present in the Alzheimer's brain as well, literally, and found the gingapane proteins there, which is something else that your team is great at characterizing, the proteins created by that bacterial species. It's just work that, adds up to be incredibly exciting. As in, we have, first this paradigm where bacteria can really be driving the inflammation and neuroinflammatory processes in these conditions, and also P. gingivalis alone. And then there's more bacteria. I'm sure there's, different bacteria, but P. gingivalis alone can cause so many issues, and it's so understudied in that capacity. And the fact that it's just right there in people's mouths most of the time is, even in people who have healthy teeth, but it, can change. It's just, think about, there's just so many therapeutic possibilities. There's so many just interesting trends off that alone. So yeah, it makes me excited just to hear you talk about it. So yes. Well, cool. So basically, when you talk about the changes in iron metabolism and coagulation and amyloid, you've gone over that. Do you want to add anything more to that? How they all intertwine? They're all related. That's one of the strengths of your work.
Yes, so we do think that they are very much connected with each other. So especially with the dormancy, if we think about dormancy, we think that periodically during episodes of stress for the person, or during inflammatory onslaught, something happening, or disease, something triggering the bacteria that might be dormant, that they might come out of dormancy. And one of the things that we think triggers it might actually be iron. And obviously all sorts of other molecules that they would thrive on. And it's well known that iron is one of the molecules that bacteria actually uses in its own metabolism. So we do think everything is connected with each other. So although dormancy is obviously a very difficult thing to study, and I think the big thing to do and to look into, probably from a microbiologist point of view, is to actually resuscitate these dormant bacteria in blood samples. So some people have already looked at the sRNA, and they have actually found bacteria via other mechanisms in blood. So, but we do think everything is connected. And the fact that traditional focus, if we look at Parkinson's just as an example, and if we look at figure 6, traditionally the focus is more being like a synuclein focus, or centric, or a neurocentric approach that people follow. But we think we should rather shift the focus to more prominently include the role of lifestyle, environmental factors, iron levels, and the result of all of these inflammatory biomarkers from bacteria as well, the inflammagens, as well as then the innate immune activation, because everything, as you can see in figure 6, we think everything is connected, and you cannot just focus on the brain, perhaps to eliminate whatever's wrong in the brain at that stage, whether you're looking at Alzheimer's or Parkinson's. I think in order to address these conditions that are just, it's just the numbers of people suffering from these conditions are just mind-boggling. So somewhere we're missing something. So I think instead of focusing on getting the disease sorted out when it's already there, we might start long before the disease has progressed to the actual disease where it is now, where the people are just suffering so much. So I think everything, yes, definitely is connected with the innate immune activation and inflammagens in the blood.
Right. I couldn't agree with you more on that, which is that my interest when it comes to developing treatment for chronic inflammatory disease is if you can understand, as you have, what's driving the inflammation, then you can try to prevent those factors from really becoming problematic in the first place. And what your work also shows is that by being connected, if you can mitigate one factor, perhaps you can mitigate the cascades that then flow on from that to the next to cause, you know, an inflammatory environment that really increasingly trends a person towards disease. So early intervention and prevention is just, it's a dream. Medicine doesn't do that as much as it does now, but if we could enter that kind of paradigm, it's really exciting that we might be able to prevent diseases and, you know, even sometimes what are called aging processes, just healthier people as they get older. So there's just real excitement in that. And, you know, one thing I wanted to mention is that when you talk about factors where you talk about the fact that stress or something else from a person's environment can, you know, kind of knock them down in a way in simple terms and allow the bacteria that are sometimes dormant to become more active and, you know, express inflammagens and begin the coagulation and clotting processes that are, you know, amyloid dysregulation, is that viral infection can be one of those things. And that's what's interesting is, and I find that interesting is, let's say you can come up with a model where someone sustains a viral infection, like a herpes infection, Epstein-Barr infection or something like that. And the virus, most viruses knock down the immune response in order to survive. They, you know, knock down T cell function or, you know, dysregulate immunity. So under those conditions, that might actually allow bacteria in the blood and in the mouth and in the gut to act more along the lines of what you're talking about. So basically a viral infection, a persistent viral infection could support the ability of bacteria to drive, you know, inflammation in the way you're talking about, and vice versa in a way, which is that if someone has, you know, a bacterial dysbiosis in simple terms in blood the way you're describing, and then they get a viral infection, they might be less able to clear that infection because the blood and the immune response is not in good shape. And that even comes down to something like getting the coronavirus now or COVID. You know, if we could better fix and understand the inflammatory processes tied to bacteria in someone's blood, would that change what the virus does? And so I find that really interesting, is how your work actually also supports the involvement of viruses in disease, even though your papers don't focus on that as much. But yeah.
Absolutely. Absolutely.
Cool. Well, yeah. So I guess with your work, there's such beautiful images, just very cool microscopy, and it really does make the research come alive. I think it's true when you hear someone say an image is worth 1,000 words or something like that. I think that's true of your case. And so can you explain a little bit more of the methods you use and sort of the way you're able to get these beautiful images of what's happening in blood?
Absolutely. So we use the scanning electron microscope to look at the structure of fibrinogen, as well as platelets and red blood cells, and then also obviously confocal fluorescent microscopy, as some of the figures that we've just shown previously, where we look at the antibody amyloid stains and more from just binding at specific places. But my first love is still the scanning electron microscope. And if you look at figure 7A, there's an example of a scanning electron microscope of a blood smear from a healthy individual. And then in 2016, this is actually a micrograph from a 2016 paper of some of our raw data. We also, if you look at figure 7B, is where we took the same individual and we added interleukin 6 at low levels, where you would have chronic low-grade inflammation with chronic levels, heightened levels or increased levels of interleukin 6. And there in figure 7B, you could immediately see what will happen when you add interleukin 6 to a control healthy blood sample, where the platelets, red blood cells seem to be fine, but the platelets just are overactivated and hyperactivated, and they grab hold of the red blood cells and become activated during this addition of the molecules. But then we also thought, well, if that can happen when you add one molecule to a blood sample, a healthy blood sample, what does it look like in various diseases? And figure 7C shows just some of the diseases that we've been looking at, including rheumatoid arthritis and diabetes, where the red blood cells themselves change their shape. And those little funny shapes that you see there is the first stages of eruptosis, which we've also been studying. Now, eruptosis is a term that Professor Lang from Germany, he's published nearly hundreds of papers, I think, on the topic. He first described eruptosis, which is a type of programmed cell death like apoptosis, but because red blood cells do not have a nucleus, the process is a little bit different, but comes down to the same process. So eruptosis is basically a programmed cell death for red blood cells, which in the presence of these inflammagens in circulation causes the red blood cells to go into these funny shapes, and also obviously platelet hyperactivation. And then figure 8 is where we look at clots, the fibrin clots, where we take plasma from a healthy or from a diseased individual. An example of a healthy individual is shown in figure A and B, where we add thrombin to activate the coagulation process to make the clot. And then figure C is of a Parkinson's individual, where you could see this hypercoagulated, thick, matted, dense clots with little fibrin fibers visible, but just more these plates of amyloid that you can see in figure C. But then we've also studied various molecules that can act as an amyloid mopping agent. And figure D is then the same patient in figure C, where we added LPS binding protein, which then changed the morphology back to a more healthy looking clot. So we published that as well in a few publications in scientific reports in cardiovascular diabetology, as well as PLOS ONE.
Wow. It's so cool. Yes. And just to summarize a little bit, the fibrinogen in simple terms. So basically the fibrinogen coagulates accumulates with sort of the inflammatory products from the bacteria. The bacteria are also themselves trapped in the fibrinogen or not?
Not typically. What you would typically find is so fibrinogen is usually soluble in circulation. And then when there's inflammagens present, and it can be also cytokines or LPSs or other bacterial inflammagens, they attach to the soluble fibrinogen, and then they form little clotlets, spontaneous clotlets in circulation. But when you add thrombin to induce the soluble fibrinogen to become insoluble fibrin, they form this little net. And Professor Weisel from the USA has worked for a long time on all of these coagulation processes. And he was one of the first people to actually also show these blood clots, fibrin nets. So during, when you are inflammatory, all of these circulating inflammatory molecules, of which LPSs and gingerpains might be some of the molecules in circulation, then binds to the fibrinogen, makes the soluble fibrinogen in certain areas insoluble or so that these clotlets form. And when you add thrombin to a sample from a Parkinson's or Alzheimer's or any other inflammatory condition, diabetes, then it clots in this plates rather than the nice nets.
Wow. So interesting. I assume that you think that this plays a role in heart disease and clotting and atherosclerosis and stroke.
Absolutely, yes, because in many instances, these hypercoagulable areas do form more spontaneously in an individual who does have all of these atherosclerotic plaques, and where some of these areas, the hyperclottable areas, come into contact with the atherosclerotic plaques and with the collagen when the shear force rips little pieces from the atherosclerotic plaques from the endothelial cells, exposing the collagen and then triggering the coagulation process. So you might find these little clotted areas more spontaneously forming in a inflammatory person with atherosclerosis And then that might definitely contribute to heart attacks as well as strokes, as well as embolisms in the lungs, or even deep vein thrombosis.
Definitely. It certainly makes more sense than the clot forming randomly for reasons that are mysterious. That's what I love with the topic of infection, is once you understand infectious processes in the body, it helps explain the mechanisms by which things can develop much more than just sometimes randomness, which is, assumed like, oh, someone just got a bad blood clot. It's like, well, I bet there was a reason. So that's a really interesting series of things. I can't help asking, when you talk about collagen being involved in that, there's a couple disorders that I'm interested in, Ehlers-Danlos syndrome, for example, where people have really dysregulated collagen to a point. Obviously, these processes can impact collagen as well, bacterial activity.
Yes, I'm sure it will. I haven't studied that disease myself. However, if collagen comes out of damaged endothelial cells, or if the collagen itself is dysregulated, I'm sure it will trigger a amyloid type of fibrinogen. Do you know if in these conditions hypercoagulability is a dilemma, or hyperclotting?
A bit. They're really understudied, and they're certainly understudied when it comes to infection. So there's very little work on that. And in fact, I might get some blood samples from patients with that diagnosis, and I'd just love to send them to you. we should check. But yeah, but yeah, no, so that's another interest. But yeah, well, anyway, though, going back to you. So I guess if you had to pick what has fascinated you most about about your research personally, you can see it basically blows my mind, but if you had to choose.
I think the most fascinating thing for me was when we discovered that this really minuscule concentrations of LPS and other bacterial inflammagens could actually induce this domino effect of soluble fibrinogen in the presence of thrombin to trigger extensive pockets of amyloid areas with misfolding protein aggregates, and that these in part may be the cause of systemic hypercoagulation. At first, when we started looking at the concentrations of LPS that might cause it, I mentioned Douglas quite a few times now, but him being a biochemist, he was from that time in Manchester, Skyping and telling me it can't be these small so minute concentrations of LPS. But when we started to try to figure out how it actually does, that's actually one of the discoveries that is really the nicest thing. And we've now done this experiments hundreds of times, making up LPS from scratch at levels as low as 0.03 nanograms per liter, which can trigger a change in fibrin and making it amyloid. So that, I think, is the most amazing, fascinating thing that we discovered.
It really is. And one of the things that's most interesting about that is that a lot of times when I try to explain to people that, bacterial activity or organism activity in blood or other tissue can contribute to a disease, it's a well-intentioned person, but an MD or a doctor will say something like, but we ran a white blood cell panel, or we ran a, just regular, blood test. And it's like, well, look, and it's a little bit expected that if something's happening in the blood, it's going to be happening in just like a major, the blood will be full of whatever the problem is, or it's just going to be exploding with the issue. But more and more in chronic disease, I see a trend where the factors that drive inflammatory processes can actually be very subtle and small, but the cascade of what they're able to set off becomes large. And so we are not necessarily looking for something extremely obvious. We actually have to look carefully and with intent, which is what you guys do, which is really interesting. So yeah. Well, finally, then, I guess overall, generally speaking, the role that persistent bacteria, viruses, and other pathogens play in chronic inflammatory disease, what do you think? I mean, I can tell where you're going to go, but, what's your overall takeaway with that?
Well, I think that these pathogens play a much more prominent role, as you mentioned, than most clinicians and even researchers can imagine. And examples of this we have shown in figure 9 and figure 10 that you have. We recently also published papers in seminars on thrombosis and hemostasis, where we actually do discuss the role of viruses also, obviously, and also bacteria, and how they activate platelets and how this might lead to thrombocytopenia. And you can just see that there's in those two figures that we try to show that, you know, the toxins from all of these molecules, the inflammagens, as well as from the viruses itself, when they bind to receptors, can just cause havoc to the coagulation process. And if you look at figure 10, what was so fascinating to me is the amount of receptors that platelets and endothelial cells have. You know, it's just mind-boggling to think how many receptors there are where these circulating inflammagens can just go and attach to.
That is really interesting. Yes. And in terms of the coronavirus, you know, COVID-19, do you think that's why we're seeing that a lot of people are having coagulation and blood clot and stroke type issues?
Absolutely, and just currently we've been working on COVID-19, myself and Douglas Kell, and also studying clotting in COVID-19 together with a really wonderful clinician, Dr. Jaco Loubser from the Mediclinic of Stellenbosch, and Dr. Jan Steenkamp, who's a hemopathologist from a pathology laboratory, PathCare, in Stellenbosch. And we have just submitted the paper actually, and you can see the figure 11 that is one of the figures in our paper. It's a review at this stage. We're also working currently on actual samples to try to clarify what's happening. So in this review that we've just submitted, we discuss how vascular implications of COVID-19 can actually relate to circulating inflammatory biomarkers and endothelial damage and erythrocytes and platelets dysfunction. And here we basically, in this review, focus specifically on fibrinogen and D-dimer, von Willebrand factor, as well as P-selectin, that are well-known circulating molecules. And if you look at figure 11, we try to show that during the various stages of the disease progression during COVID-19, these markers may either be within healthy levels, or they may be upregulated, or they might eventually be depleted. So most significant is that patients need to be treated, we think, quite early in the disease. In figure 11, you can see our green arrow, the optimal time of intervention. So at that stage, when the patient comes in with abnormal clotting, fibrinogen levels are typically high, von Willebrand factors are high, P-selectin factors are high, but interestingly, Dr. Loubser found that D-dimer were low or they're normal levels of D-dimer. However, as the disease progresses and the virus attaches more to the endothelial cells and the lungs, then it shifts to a bleeding and thrombocytopenia where the fibrinogen is depleted, von Willebrand factor is depleted, P-selectin is again now more upregulated, but then interestingly now in the later stage, D-dimer is also upregulated because obviously the fibrinogen that's formed and the fibrin that's formed, which we think might be amyloid, are actually now breaking down to form the D-dimer. So there's absolute need for personalized medicine. So obviously one must note that this is possibly not a one fit for all patients. You do have the patients that start off with a bleeding tendency before they develop COVID. So obviously there's no one fit and say you should drink platelet medicine that might influence platelet reactivity for everyone. However, we do think that the papers that we see that's been published talk about a hypercoagulable abnormal clotting state, but they also talk about people going into bleeding and thrombocytopenia. But that cohort might be after they already have had the abnormal clotting and the clots have already formed in the lungs, and then they get this dysregulation and bleeding and thrombocytopenia because of the fact that during the disease progression, these molecules are simply depleted. So definitely, it plays a crucial role in all of the pathology of COVID-19 as well.
Very interesting. Yes, I can already think of a couple therapeutic interventions you could do there. Exactly. If you could just characterize exactly from patient to patient what's better happening in the blood, that is very exciting. Wow, I don't know how you get all this work done. You study so many topics. Do you have any other interesting things you're working on, or did we hit the high points?
I think we hit the high points. I think that is what we've been working on this past few weeks. Yes.
Great. And then I suppose if there are young researchers that are interested in this topic, what advice do you have for them?
I think the most important advice I can give anyone, any young researcher, is I think the most important lesson that I have learned over the years is that the evidence is what it is. You should never decide before you do a study what you will find. I think, and it's most important to remember that the handbook and what you've been studying all along and the known dogma could be wrong. And the most important significant discoveries that we can mention over the decades was once thought to be wrong. So I think we should just look at the evidence in front of you and then formulate your hypothesis on what you see. It's very difficult to, if you've got the set mind to what you're supposed to see and you don't see it, to go in search of something that might not be there, but the evidence might be right in front of you.
I agree. I couldn't agree more. Just approach the topic with an open mind and think independently. That's great. Very cool. Well, Resia, it's been wonderful to talk to you. As you know, I'm trying to get you samples to study from patients with the condition myalgic encephalomyelitis, which is sometimes called chronic fatigue syndrome, and also maybe patients with what's called post-treatment Lyme disease involving Borrelia. And when I get that, I am so excited for you guys to take a look because I doubt that there's nothing going on.
Absolutely. So it's a pleasure to look at those because I really agree with you. I think coagulation and bacterial influences, dormancy might be something else to study. However, inflammagens in the blood affecting the coagulation, platelets, and red blood cell structure, definitely it's something to study in the future.
Great. Well, that's wonderful, Resia. All right. Well, thank you so much for your time and keep up the great work. And I'm going to follow what you're doing with COVID and we will stay in touch. And thank you.
Absolutely. And thank you for your wonderful work as well. You've always been an inspiration to myself and Doug.
Oh, that's so cool. All right. Thanks, Resia. All right. Take care for now. Bye.
Bye.
Bye.